The European Union on Tuesday approved the world's first medicine based on CRISPR gene editing technology to treat 2 blood disorders, following the US and UK in a milestone for the revolutionary technology and a breakthrough in the fight against human genetic disease. CRISPR gene editing works as a precise pair of genetic scissors. By allowing scientists to alter specific parts of the human genome, CRISPR opens up a way to correct or work around diseases with genetic components like sickle cell or beta thalassemia. Inserting stable changes to genes in a living person is no easy feat, and while Casgevy appears to successfully edit DNA inside the human body, it doesn't actually address the hemoglobin production issues causing the 2 conditions. Instead, Casgevy implements a genetic workaround by targeting genes responsible for producing a different kind of hemoglobin that normally stops shortly after a person is born. This genetic workaround triggers the production of fetal hemoglobin in the body, which is sufficient to help restore normal functioning to red blood cells and allow them to keep their healthy, disc-like shape despite the absence of properly formed adult hemoglobin. While not technically a cure for the disorders, in practice it can function as one, and clinical trials suggest the one-off treatment helps relieve symptoms of disease and could free patients from the need for bone marrow transplants or regular blood transfusions. While the treatment is administered once, the therapeutic process can take months, including laboratory work to modify blood cells and a recovery period for patients in hospital after the modified cells are infused into them. Long-term studies will be needed to see how the treatment fares over time. The regulatory green light for Casgevy in Europe comes months after the European Medicines Agency recommended it for approval in December. The bloc is now one of several jurisdictions to have authorized the treatment for the 2 conditions since the UK became the first country in the world to greenlight a CRISPR-based therapy last November. The Food and Drug Administration in the US authorized Casgevy for sickle cell in December and for beta thalassemia in January. CRISPR stands for clustered regularly interspaced short palindromic repeats. It is a powerful gene-editing technology derived from the immune systems of bacteria. It offers scientists a previously unimaginable degree of precision when manipulating DNA, life's instruction manual outlining how every cell in our body works. The tool has already transformed the life sciences in the short time since its discovery just over a decade ago. Its discoverers won the 2020 Nobel Prize in Chemistry, and has opened up an array of possibilities ranging from correcting faulty genes in medicine, producing new materials, tweaking or transplanting genes across species to enhance or introduce new traits for things like disease resistance or crop yield.